All right, guys, welcome back. Let's go ahead and pick up where we left off, and in many aspects, duplicate what we've talked about with the digestive system. Like we do in lab here, I have selected an absolutely outstanding video. This is an AI driven video that I've used for several years now that is really, really well done. We talk about, again, in lecture, it's anatomy with more physiology, how things work. In lab is much more anatomy, learning the parts, learning the organs, the accessory structures, etc. But this video does a beautiful job of both the anatomy and some of the enzymes and what's occurring at various levels of the digestive system. I think it's going to be a really nice complement, and again, intended by very intentional design to supplement lecture and bullet points for those of you that learn more visually. So that's why we design and purposefully do these videos, okay? So we'll go ahead and roll through this video, especially when we get to the stomach and some of the layers of enzymes. It gets a little bit in depth. Don't get overwhelmed by that. I'm going to tell you exactly which enzymes you need to know. This video is outstanding, but it does go a little bit deeper than we will be going here in Bio 211 at MTC, okay? So let's watch the video and then we'll reconvene. See you guys in a few minutes.
The human digestive system is very complex and has evolved over millions of years. It basically consists of the rectum, the large intestine, the small intestine, the pancreas, the stomach, also called gaster and ventriculus, and the liver with the gallbladder. The esophagus is also part of this system, as well as various salivary glands near the mouth. First, the food is broken up in the mouth by the teeth and then mixed with saliva with the help of the salivary glands. Saliva contains a digestive enzyme called amylase that already begins to digest carbohydrates in the mouth. It splits carbohydrates into smaller units. The ball-like mixture of food with saliva, also known as bolus, is pushed into the throat by the tongue, and finally into the esophagus, which propels the bolus to the stomach. The esophageal lumen, that is, the opening inside the esophagus, is very flexible, which allows boluses of different sizes to be transported. The esophagus consists of several layers. These layers occur throughout the entire digestive tract. The two outer muscle layers are responsible for peristalsis. Through these two muscles, the bolus can be transported from the mouth to the stomach, even if the person is standing on his head. Stomach is often divided into 6 areas. The stomach is composed of a similar structure to the esophagus. It has a longitudinal muscle layer on the outside. Underneath, we can find circular muscle fibers. In addition to this, there is an oblique muscle layer overlaying the mucosa. On the inside, there are rugae that allow the stomach to enlarge when food is consumed. The stomach wall contains gastric glands. They produce mucus, which is able to protect the stomach wall from the secreted gastric acid. Gastric acid is produced by simply smelling or seeing food, but also spices, and the stretching of the stomach causes the secretion, that is, the release of gastric acid. About 1 to 2 liters of gastric juice are produced per day. Since the esophagus does not have a protective mucus layer like the stomach and esophagus are separated by a sphincter. It relaxes when a bolus is pushed from the esophagus into the stomach and then contracts to prevent acid and food from going back up. Gastric juice consists, among other things, of hydrochloric acid, the enzyme pepsin, the intrinsic factor, and lipase for the digestion of fat. In addition to nutrients, food also contains bacteria that can damage the body. The components of hydrochloric acid are able to destroy harmful bacteria. In addition, hydrochloric acid converts pepsinogen, also released by the gastric glands, into pepsin. Pepsin is able to break down proteins in the stomach. For a vitamin B12 absorption in the small intestine, the intrinsic factor is needed, which is produced by the gastric glands. The vitamin must combine with intrinsic factor, then it can be absorbed later by the small intestine. Vitamin B12 helps keep the body's nerve and blood cells healthy and helps make DNA. It also contains gastric lipase, an acid-resistant enzyme for fat digestion. In the stomach, gastric lipase splits a triglyceride into a free fatty acid and a diglyceride, whereby only the free fatty acid can be absorbed by the body. More effective fat digestion takes place in the small intestine. Through gastric juice and stomach movements, which take place approximately every 20 seconds, the individual boluses are mixed to a semi-fluid mass of partly digested food, the so-called chyme. The chyme cannot enter the duodenum at first because there is a sphincter at the stomach exit. The pyloric sphincter resembles the esophageal sphincter. The pyloric sphincter opens only a few millimeters, so that larger pieces remain inside the stomach. In the first section of the small intestine, the duodenum, bile, and pancreatic secretions are mixed with the chyme via the ampulla of Vater. Pancreatic juice contains numerous digestive proenzymes and enzymes. In order for these to do their job, a higher pH value than that in the stomach is necessary. For this reason, pancreatic juice contains sodium hydrogen carbonate. Hydrogen carbonate is able to neutralize the acid in the chyme and thus produce the optimum pH value of 7 or 8. Pancreatic juice also contains proenzymes. It is only through enterokinase, released by the duodenum wall, that the proenzyme trypsinogen becomes trypsin, which can split proteins and activate other trypsinogens. We also find alpha amylase, which we had already found in the mouth. It now does the rest regarding the splitting of carbohydrates, which it converts into maltose and isomaltose. Furthermore, pancreatic lipase is able to split triglycerides into 2 free fatty acids. The gastric lipase, as we have seen before, can produce only 1 free fatty acid. The pancreatic lipase can cleave triglycerides excellently because the bile breaks the fats down into tiny droplets. This is called emulsification. Numerous other enzymes are part of pancreatic juice, but these will not be explained in detail here. Bile is produced by the liver cells and transported to the gallbladder. The bile is stored in the gallbladder and finally added to the food in the duodenum via the ampulla of Vater. The pancreas also releases juice via the ampulla of Vater. The small intestine consists of three sections: duodenum, jejunum, and ileum. The ileum continues into the large intestine in the right lower abdomen. The duodenum and the jejunum have circular folds to increase the contact surface with the food. These circular folds extend about 1 centimeter into the lumen of the small intestine. These folds are covered with small finger-like projections called villi. Villi increase the surface considerably. Villi are about 1 millimeter long. Each villus contains blood capillaries and a lymphatic capillary called lacteal, which we will see later. The nutrients, marked green here, are absorbed by the villus and transferred to the blood capillaries. Some nutrients, such as glucose, do not require a carrier. They are transported freely in the bloodstream. Other nutrients, such as iron, require transport proteins like transferrin. Fats are transported by chylomicrons, which are lipoproteins. The triglycerides to be transported are virtually enclosed in the lipoprotein. Chylomicrons and triglycerides are then transported through the lacteal of the villus. Each villus is covered by even smaller microvilli. They multiply the intestinal surface considerably. The microvilli absorb nutrients and transport them to the inside. The last part of the small intestine is the ileum. It does not have, unlike the duodenum and jejunum, circular folds. The ileum absorbs electrolytes, such as calcium, for building bones, hair, and teeth. Trace elements, such as zinc, for sperm production and the immune system. Vitamins, such as B12, for the formation and maturation of red blood cells. And remaining bile acid, which is transported back to the liver via the bloodstream. As with the esophagus, the food is transported by peristalsis. In contrast, segmentation contractions serve to mix the chyme, which is shown here in yellow and red, to make it easier to see the mixing process. The large intestine is thicker than the small intestine. It is about 1 meter long and surrounds the small intestine. The small intestine is connected to the large intestine via the Bowhem's valve. It opens when chyme is to pass from the small intestine to the large intestine. The large intestine does not have any villi like the small intestine because most digestible substances have already been absorbed in the small intestine. However, the large intestine has an estimated 100 billion bacteria inside. They are important for many other tasks, such as the production of vitamins and the decomposition of fiber for the body's energy production. Many of these bacteria are an important part of the immune system by killing harmful germs. Through peristalsis, the chyme is transported from the ascending colon to the transverse colon to the descending colon. On its way through the large intestine, water is removed from the chyme. Furthermore, mucus is added for proper excretion of waste. Substances that cannot be absorbed through the small intestine or the large intestine remain in the rectum and are finally excreted through the anus.
So, like I said, a really nice video on the digestive system, really well done, and was much cooler actually a couple years ago, 3-4 years ago, before AI became so prominently a part of our existence. At the time, it was almost sci-fi space age type stuff. But I love the graphics. It does a great job. Like I cautioned you guys and shared with you, please don't get caught up in all the levels of enzymes and interaction. A lot of that is much more in depth than we're going to go at the college in Bio 211. Okay. Those would be more of a grad school level type class. But overall, worth watching, and just glad that I have that to share with you guys. Let's go ahead then and let's work on yesterday, on Monday. I emailed to you guys a digestive system handout, very similar to the respiratory handout that we did 2 weeks ago. And by the way, at the conclusion of lab today, I'm going to talk a little bit about the coming quiz, etc., and how to prepare additionally. So just make sure you stick with me here. But that respiratory handout, as I shared with you when we met 2 weeks ago on Tuesday before spring break, is going to be important for studying, as will this digestive study guide. So you can see that there are a number of things to be labeled after going through lecture with me previously today, as well as now seeing this video and recapping and touching on the various components of the digestive system. Many of them should be pretty obvious to you, but what we're going to do is we're going to go ahead and take a break, and I'm going to let you guys have a breakout session where you can start to research, use your phones, your computer, whatever it takes to start to label this handout so it can become a study guide for your next lab quiz, which will be 1 week from today, lab quiz 3. Okay, so let's go ahead. It is, let's call it 4:15. Let's come back in 20 minutes from now. Okay, so that would put us at 4:35. We'll come back 4:35, work on your labeling of the digestive system handout, and we'll see you guys back after the breakout session. Okay, guys. Hopefully you were able to fill some of them out. Hopefully many of them made sense to you after what we've covered both in lecture and in that overview video that we just saw. So let's go ahead and take a look at the handout. And I want to do as I did with the respiratory handout that we created, the study guide for the respiratory system. Kind of steer you in on the things that I want you to prioritize when it comes time for studying for lab quiz number 3. Okay, so let's start over on the right-hand side of the page with the letter A at the very top. Can you guys confirm to me, please? Someone, you can even just drop it in the text if you want or whatever. But are your arrows labeled, or do they have letters next to them? Oops.
Yes, it does.
Okay, there we go. Now I see it. My chat was frozen. Sorry. Thank you so much. Okay, I didn't want to start calling off letters if you guys didn't have them. Sometimes our copies differ a little bit. Okay, so let's start over in the top on the right-hand side, letter A then. All right, so that is pointing to the salivary gland called the parotid gland. Okay, these that I'm about to call out here for you are the ones that you absolutely want to be familiar with for the lab quiz. Okay. So,
excuse me, Dr. Fine.
Yes.
The picture for the picture is not showing on the screen.
No, you have the picture in front of you.
Okay.
Yep.
Okay.
I'm just calling them out. Okay. So because you guys should have them labeled anyway, so we're going to proceed. All right. So the letter A is that parotid gland. Okay. And then the letter B right underneath it is the sublingual salivary gland, that one that's under the tongue that we talked about. So definitely for the lab quiz, know those first two letters A and B, parotid salivary gland and the sublingual salivary gland. All right, we're going to skip over the letter E and we're going to continue down the page on the right-hand side. The letter combination B-E, B as in boy, E as in Edward, is the stomach. And again, a lot of these we'll be seeing in the PowerPoint slides coming up here, but I just want to get this locked down for you first. So B-E is the stomach. You'll definitely want to know the stomach. Right underneath that, C as in Charlie, D as in David. CD is outlined to illustrate that it's in that retroperitoneal cavity back behind what we can see, but that is pointing to the pancreas. Okay, and we'll be talking a lot about the pancreas on Thursday over in lecture. It's a major player secreting enzymes. All right, underneath the pancreas, continuing down that right side of the page, C is in Charlie, E is in Edward. CE is the transverse colon. Transverse colon. Right underneath that is another segment of the large intestine or colon, DE, and that is called the descending colon. And it's nice that the anatomists gave us pretty easy names when it comes to the sections of the large intestine / colon. For actually, if you remember the direction that the waste product at this point is traveling, you'll know the region name. And we'll talk about that coming up. We're going to skip ABC underneath that. But then what I would like you to do, and let me see here, I'm going to stop sharing so that I can blow this up. Okay. All right. Can you see on my page here where I drew in this arrow?
Yes.
All right. So that's the only draw in arrow that I need for you to do. And that's right down at the bottom of the descending colon where it makes the turn is called the sigmoid colon. Sigmoid. Okay. That's the only one that I need for you guys to draw an extra arrow in that wasn't included on this worksheet, but it's going to be important for you to know for the lab quiz. All right, then let's go ahead and cross over to the left-hand side of the page. We're going to skip the letter C, and we're going to move down to D as in dog. And that is, of course, I hope everyone would know, that is your esophagus. That smooth muscle tube carrying bolus to the stomach. As we can continue down the left-hand side of the page, the letter combination A as in Adam, B as in boy, AB is important to know that that is the right lobe of the liver. Okay, the right lobe of the liver. And we're going to see all four lobes in a few minutes. And then continuing downward on the left-hand side of the page, we're going to skip AC and go down to AD. A is in Adam, D is in dog. And that is going to be another part of the large intestine called the ascending colon. Okay, so with the ascending colon, which goes up from the small intestine, then the transverse colon, cutting laterally across the abdominal cavity, and then the descending colon, of course, descending, and then the sigmoid colon before the large intestine becomes the rectum. All right, and that's it. The last three on the left-hand side, we're going to ignore. So just to double check, you guys should have labeled. Well, let's go down the left-hand side that we just talked about. The letter D you should have labeled, AB and AD. Those three should be labeled on the left-hand side of the page. On the right-hand side of the page, you should have labels for A, B, BE, CD and CE, DE, and then we drew in an arrow for the sigmoid colon. Does that, and anyone need any of those? Anyone missing any of those? Those are the ones that you're going to need. Okay, terrific. All right, so keep that, put that with your respiratory study guide sheet. Again, this is a point of emphasis for anatomic parts related to the digestive system, to the alimentary canal that you are absolutely going to want to be able to identify and recognize for lab quiz number 3 next week. At this point, let's go ahead and proceed on to the slides, which of course you can always access on D2L. They're under the content section. And we're going to pick up, if you remember last time, we finished with the respiratory system, and specifically we're talking about spirometry, right? And then we stopped right before we got to the digestive system. So we're going to pick up there and go through the slides related to the digestive system for lab quiz number 3. And again, my slides are always a point of emphasis that you should be studying to be successful on the lab quiz. All right, so first of all, we know from lecture, we talked a great deal about teeth being accessory digestive system organs. There are, of course, as all of us know, different types of teeth. And in the front, we have the more flat teeth that are called incisor teeth. And then as we move around and toward the back of our jawline, we have our pointy, four, two on the top, two on the bottom. The most pointy teeth are called our canine teeth. The incisors and canine teeth are primarily used for ripping or shredding food. If we continue moving around the jawline from the front, past the canines, we have premolars, which look a lot like molars, except they're just a little bit smaller in size, typically. And then, of course, as we move to the posterior of our jaw, top and bottom, we have our molars, which are the largest teeth, okay? And our molars and premolars are really responsible more as mashing and grinding to convert food into a bolus, a paste-like substance, okay? So for the lab quiz, definitely be familiar with these four types of teeth, okay? The four primary types. All right, let's continue. And again, you can use this slide presentation as your study guide, of course. All right, then we get into the actual parts of the tooth itself, okay? What's inside of the tooth, those of you going into the dental field will certainly get a lot more exposure to this. But those of you like me in healthcare, this is probably going to be the only level of learning you're going to have related to the inner functions or inner components of a tooth. The outermost surface, the white, hard, very firm portion of our tooth is the enamel, as the slide shows. And then as we move inward, then we have the dentin. Is in this illustration, it's colored tan. In real life, of course, it's not. But the dentin is more of like connective tissue that's kind of holding that enamel, anchoring it into place. And then if we move deeper into the inner components of the tooth itself, we get into the pulp. And it's here in the pulp, which is colored red here. Here in the pulp is where we have the vascularization. We have blood flow being brought, waste product being removed. This is where the nerves, where all those roots that become inflamed, if we need a root canal, you know, that extensive, we know that the nerves of a tooth, anyone who's ever had a broken tooth or a root canal knows how sensitive our nerves of our tooth can be. Those nerves, along with the blood vessels, are all in this pulp area of our tooth, okay? So for the lab quiz related to the teeth, I would really be familiar with the pulp area and the enamel area, okay? There's really only those three. Around that, of course, is the bone where the tooth is embedded, and then the gums, the fleshy, vascularized portion of tissue that overlaps the bone and also works to hold the tooth into place. We're not going to worry in Bio 211 about the gums and the bone. These three parts of the tooth are what I need you to know for the lab quiz, and specifically make sure you're familiar with the pulp, this inner vascular area that's highly filled with nerves, as well as the enamel, the white outer, very, very rigid, firm surface. You can think of all the things that you've chewed in your lifetime and how that enamel, for the most part, doesn't break. It's not foolproof, but it's pretty good at what it does. All right, continuing. Let's move to the salivary glands again, and this should be a recap of what we've talked about, okay? We have that parotid gland. There's the three different salivary glands. The parotid gland on either side, again, immediately anterior to the ears on either side, lateral side of the cheek area of the face, okay? Largest of the salivary glands, the parotid. Remember, I told you on your diagram that we filled out that you would need to know that one, okay? Then we have the submandibular salivary gland, which lies, just like I said in lecture, right under the mandible. And then we have the sublingual salivary gland. And like I said over in lecture, anytime we hear sublingual, we're talking about underneath the tongue. Okay, so those are our three salivary glands. Be familiar with the two that we highlighted over in when we were just filling out our worksheet, our study guide, the parotid and the sublingual under the tongue. Okay, those are the two that you really want to emphasize for the lab quiz part. All right, then to the stomach. And again, we left off over in lecture prior to getting to the stomach. That'll be the first area that we pick up with. And you'll notice that there's some strange spellings here. Don't worry about the spelling like of esophagus here. Over in Europe, in certain areas, they put an O in front of esophagus. So don't just spell ours the normal way, okay? Everything else is accurate, okay? All right, but I mentioned previously when we talked about that cardiac sphincter where the esophagus terminates and meets with the stomach, that it's not truly cardiac when it comes to heart, but the cardiac region, or sometimes called cardia region, is that first portion of the stomach that the esophagus physically turns into, okay? The alimentary canal. So that's one of the four regions of the heart, is the cardiac region, right where the esophagus melds into. Up at the top, the most superior aspect of the stomach is called the fundus, as you can see. And then the largest of the four portions by far is called the body of the stomach, colored here in yellow. And then if we're following the pathway of the alimentary canal and bolus progressing through the alimentary canal, the last segment of the stomach before it would pass into the duodenum, the first section of the small intestine, is called the pylorus. Okay, so the cardiac region, the fundus at the very top, the body is the largest, and then the pylorus is, again, if you're traveling the path of bolus, the most inferior and last segment of the four regions of the stomach before entering into the small intestine. Okay, so for your lab quiz related to the stomach, I would definitely be familiar with both the fundus and the pylorus. Okay, for the lab quiz, specifically know that fundus and pylorus, those two regions. And we'll be talking more as we progress again in lecture on Thursday about the pylorus. Then the next sphincter that we're going to have, we had the cardiac sphincter right here by the cardiac region. We have the pyloric sphincter, which is that valve-like opening that's going to let the chyme at this point pass out of the stomach into the first segment of the small intestine. So the sphincters attached to the stomach are actually named after the region of the stomach that is the entry and exit point, the cardiac sphincter and then the pyloric sphincter. All right, so know this pyloric sphincter as it says here, okay? And again, we're going to be covering this in lecture. We didn't get this far in lecture yet, but here in lab, know for the lab quiz, the pyloric sphincter. All right, let's continue. All right, so the stomach also has what are referred to as curvatures, okay? And this is just the physical shape, the outer aspects of the stomach. They're real easy. There's only two of them, as we can see, and they're real easy to remember because they make sense. Finally, the anatomists did us a favor and named something that was very common sense-ish. Okay, this entire loop here on this entire aspect on the dorsal side of the stomach is referred to as the greater curvature, which is also much greater in size than the lesser curvature, which is this innermost curve, which is much lesser, smaller. Okay? So usually people do real well on the curvatures when it comes time to remembering. It's just greater and lesser, and it's based on the length. Okay? Pretty easy to remember. All right, moving and taking a look inside the stomach. And again, this is all set up, a precursor to lecture on Thursday. We'll be talking about all these things again. So you're going to hear them again in much more detail. Okay, but these ridges that we can see here, this is a cutaway of the stomach. Okay, these ridges that are so apparent in here are called rugae. Remember, in the ventricles of the heart, we called them trabeculae carneae. They were ridges that kind of looked like spaghetti on the inside lining of the endocardium. Okay, the inside raised ridges on the inner surface of the stomach is called rugae. And these rugae are there for one point and one point only. We all know that our stomach stretches, right? When our stomach is empty, it's at its smallest size. And then if we go to Shoney's and have a big lunch buffet and overeat, our stomach is going to stretch to accommodate that food volume temporarily in the stomach, right? And it's those rugae, those ridges of muscles on the inner lining of the stomach that allow the stomach to stretch and get larger. And then eventually, when the stomach empties, pull it back to size. Okay? So know these rugae. All right. Then within the inner lining of the stomach, and we're going to talk a lot about the gastric pits. Okay. This is actually on the inner surface of the stomach. And on that inner surface of the stomach, around the rugae, are millions of little indentations. They're little pits, as the name says. They're indentations. And we're going to talk about all the different cell types and what gets released from these gastric pits. But these little indentations on the inner surface of the stomach, called gastric pits, release various chemicals. The biggest one is hydrochloric acid, that acid that is released in our stomach that further physically and chemically dissolves proteins in foods and breaks our food down, right? We've all vomited at one point in our lifetime, at least, many, many times for most, right? Stomach flu, bad food, whatever it may be. We have that burning sensation, right, after you vomit. That's from hydrochloric acid being expelled from your stomach contents, okay? The hydrochloric acid is one of many things that comes out of these indentations in the stomach inner lining called gastric pits. Along with that, there are hormones, and as you can probably predict, there are a variety of enzymes that we're going to be talking about on Thursday over in lecture. They all come from these little indentations and emerge from the pits of the inner lining of the stomach out into the stomach to do what it is they're responsible for doing. This, of course, is a histology, a microscope image. All right, then we have what is called the greater omentum. And what we're seeing here is this yellow area right here, this sheet of connective tissue, okay, that is yellow or goldish color, okay, is called the greater omentum. Now, in actuality, it would continue all the way across. It's just cut and removed on one half so that you can clearly see what it's lying on top of, because here's our large intestine / colon, and here are the 3 segments of our small intestine, okay? So this greater omentum is connective tissue that lies over the large intestine, small intestine, and is anchored to the stomach. And what it's doing is it actually is there to hold these specific alimentary canal digestive system components in place. Okay. We wouldn't want our small intestine just to unravel all over the place, right? We wouldn't want our large intestine to move over the top of our stomach and start to clamp it off. Okay. So this greater omentum, this sheet that would cover the entire region here, kind of anchors everything into place and keeps things from moving around. You'll definitely want to know the greater omentum. And in fact, if it were me, I would think that this looks a lot like the quiz question. All right, now we get to those lobes of the liver that I mentioned briefly when we were doing our study guide. Okay, we labeled label AB as the right lobe of the liver. There are 4 different lobes of the liver. Okay, and remember we talked about lobes of our lungs before. Okay, various segments. Okay, like we talked about the regions of the stomach. There were 4 regions of the stomach. All right, from the front, we can only see the 2 primary lobes of the liver. Okay, so from the anterior, like we're looking over here on the left-hand side, we can see the largest lobe by far is the right lobe of the liver. And if you recall, that's what we labeled on our worksheet for you guys to definitely be familiar with. Okay. From the anterior, we can also see the left lobe of the liver. Okay, and remember, just like anytime we're looking at something, a patient, we're always looking at the patient's right and the patient's left. Okay, so this would be the patient's right. It's our left. Okay, this is the left lobe. All right, then importantly, while we're still on this anterior view, okay, from the anterior view, we have a ligament here called the falciform ligament. And it runs superiorly over the top of the liver and then down in between the left and right lobes on the anterior side. And that falciform ligament is very, very important because it, much like the greater omentum holding the intestines in our stomach into place, the falciform ligament works like an anchor, or like ropes, if you would, to hold the liver into place. Okay? You think about all the organs in our abdominal pelvic cavity. If there weren't anchors, everything could move around, right? We certainly don't want that. We'd have big problems. Okay? So we have the right lobe, left lobe, falciform ligament from the front view. When we flip that liver around and we look at the posterior view from the back at the lobe, we can see all 4 lobes of the liver for the first time. We can still see that right lobe. We can still see the left lobe over here. But now we see the 2 middle lobes, if you will, that are much smaller in size. And at the top is called the caudate lobe, as you can see with the blue arrow pointing to it. And underneath that is what's called the quadrate lobe, articulated here in purple. Okay, so still this is the posterior view. So again, the patient's right, that would be inverted. Okay, so the right lobe, the left lobe, caudate, quadrate. And for the first time here, we see the gallbladder embedded. And we'll be talking a lot more about the gallbladder coming up in lecture on Thursday as well. Works hand in hand with the liver. Okay, so for the lab quiz, know the 4 lobes of the liver, and then also know the falciform ligament. All 4 lobes plus the falciform ligament. So in other words, this whole slide. Okay, they're pretty easy, right, left. As long as you get your orientation right, you'll be fine with those. The falciform's easy. You just have to find a way to remember the caudate and quadrate. All right, and then again, the lab quiz wouldn't be a lab quiz without more microscopic histology slides, right? You've sensed a trend here. Okay, so this is a microscopic slide, a tiny, tiny, thin slice of liver tissue. Okay, the liver, of course, is a major filter among other things. And this is liver tissue. Recognize that as such. This is the image you're going to see, guys. No surprises. Okay, I'm giving you guys this one. All right, now we're back to the gallbladder and a view of our liver. All right, the posterior of the liver. Okay, I'll tell you right now, and we'll talk again more about this in lecture on Thursday to be sure. But one of the major enzymes that the liver produces and secretes when it comes to digestion is called bile. Okay, and bile is produced by the liver, as I said, and the liver can send that bile down into the lower to the small intestine. Okay, for chemical digestion. All right. But the liver also produces an abundance of bile. So the extra bile is stored and temporarily housed in the gallbladder. That's what the gallbladder does. It's a storage bin or a storage extra capacity for bile produced by the liver, ultimately injected into the small intestine for chemical digestion. Okay, we don't see the ducts that carry the bile from either the liver or the gallbladder in this drawing, but we do see the duct called the cystic duct that transports the extra bile produced by the liver into the storage area that's called the gallbladder. Okay, and that's why people can have their gallbladder removed without a significant impact typically on overall health, because it's just I don't want to say just, but it's a storage facility for extra bile. As long as their liver is intact, they have a healthy liver, having this extra storage capacity and a gallbladder isn't necessary. Okay, so that's why it can be removed typically without harm. So know this cystic duct, okay, the tubule that connects to the liver and carries that extra bile into the storage area that is the organ called the gallbladder. All right, now we get down to the pancreas, and we're going to talk a lot about the pancreas on Thursday because it is the major enzyme producer when it comes to the digestive system and chemical digestion. The pancreatic juices that are produced, those enzymes by the pancreas and dumped into the first segment of the small intestine called the duodenum, break down and chemically digest fats, proteins, and carbohydrates. They're the only enzymes that break down chemically all three. So it's a major player when it comes to enzymes and chemical digestion. Okay, so when we look at our pancreas, and remember back to Bio 210, the pancreas is technically in the abdominal cavity and partly in the retroperitoneal cavity. Remember, the retroperitoneal cavity is just behind the peritoneal cavity of the abdominal cavity. The kidneys are certainly retroperitoneal. They're outside of that serous membrane to the posterior. Parts of the pancreas are in retroperitoneal as well. Okay, so when we look at the parts of the pancreas, it depends on what source you're looking at for the parts of the pancreas as to how many breakdowns there are. Okay, they just vary depending on the author. Okay, so here for Bio 211 for the lab part of it, I'm looking here at my notes. All right, for here we're going to be most focused on the head, Okay, and it's kind of easy to remember the parts of the pancreas because they kind of flow as if this was a fish. And to me, it kind of looks like a fish a little bit. Okay, you have the head as this largest portion right here. Okay, and then as you move from there, you have the neck, and then the body, and then the tail. So those are kind of common sense, and again, very easy to remember. This uncinate, where the pancreas winds around and wraps around itself here, we can see this aspect here, the uncinate, is kind of an oddball name, okay? But fortunately, for Bio 211 here at the college, we're not going to worry about the uncinate, okay? We're going to worry about the head, the body, and the tail. So those should be pretty easy for you to remember. The head, the body, and the tail. If you think of it like a fish, it should be pretty easy for you to remember what part of the pancreas you're being asked. Okay? And then running down the surface, again, keeping with my fish analogy, and this illustration doesn't really do it justice. The one we're going to see in lecture makes it look a lot more like what I'm about to share. But this pancreatic duct, remember, a duct is nothing more than a tube that carries something from one area of the body to another. Okay, we talked about the cystic duct carrying bile from the liver to the gallbladder for storage, right? This pancreatic duct is this long tube that runs the entire length of the head, body, and tail of the pancreas, and it has all kinds of branches that come off that, to me, again, not as much in this picture, but certainly more so in others, looks a lot like the bones of a fish. So again, very easy to stick with the analogy of head, body, tail like a fish, and then the pancreatic duct being the spine of the fish bones with all the branches coming off either direction. Okay, that's where these pancreatic juices, those enzymes that are produced in the pancreas, all make their way into the pancreatic duct, and a duct doing what a duct does, carries those enzymes the length of the pancreas and ultimately dumps them into the duodenum, that first and shortest segment of the small intestine. Okay, this, for the first time now, we see the other part of the gallbladder. We don't see the cystic duct connecting in this cutaway to the liver, but this is how the gallbladder, which the cystic duct, again, is how the gallbladder receives bile from the liver to store it. The gallbladder, when it's time for the gallbladder to share that excess bile and use it for chemical digestion, it will dump the bile out of the gallbladder via the common bile duct. And then that common bile duct makes its way into the pancreas and merges together right at the entry point of the wall of the duodenum. So both bile from the liver and from the gallbladder and pancreatic juices from the pancreatic duct are all making their way into the duodenum of the small intestine at this same point. So there's a massive dumping of enzymes in the duodenum of the small intestine. Okay, for your lab quiz, like I said, know the head, body, and tail of the pancreas, know the pancreatic duct. Okay, and let me take a look just to make sure here when we're talking about the gallbladder. I want to make sure. And the only thing about for the lab quiz, the only thing you need to know about the gallbladder is that cystic duct that I showed you on the previous slide, right here. Okay? All right. So those are the pancreas parts that you need to know anatomically for the lab quiz. All right. Next, to the small intestine that we will talk about at length on Thursday. The very first segment, as we leave the stomach, and specifically that pyloric region, through the pyloric sphincter, the first segment of the three of the small intestine is the duodenum, and it is by far the shortest in length. It's only 8 to 10 or so inches long, depending on the individual. The duodenum, then, if we continue to follow, remember it's chyme now at this point, ejected from the stomach into the small intestine. And again, that duodenum is where the pancreas and the gallbladder and liver, all their ducts dump enzymes in. Okay, that duodenum again is 6, 8, 10 inches long, depending on the individual, and then it becomes the jejunum. And then the jejunum, as we continue the pathway of the chyme, becomes the ileum. And then the ileum is going to connect to the large intestine / colon, which we'll see in a minute. Okay, so the 3 sections in the order of chyme progressing through it are duodenum, then the jejunum, then the ileum. Duodenum is only inches long. The jejunum and ileum are anywhere from 10 to 15 feet each in length, all compacted and wound up tight in here, as you can see. Okay, so a significant difference in length between the jejunum and ileum compared to the duodenum. All right, so know the 3 regions of the small intestines. And again, we'll be talking more about them, so you should know them by then from multiple opportunities to hear and learn about them. All right, like the video said that we led today's lab with, the small intestine is all about absorption, that grabbing of precious nutrients, whether it be water and fluids or whether it's monomers that we've talked about, and pulling them through the wall of the small intestine into the capillaries to that hepatic portal system and shuffling those nutrients to the liver. Okay, so because absorption is the task at hand of the small intestine, we want to facilitate, we want the small intestine to be highly effective at what it does and to be able to capture and catch, latch onto as many nutrients as possible. Anything that we let go past it is waste, and we don't want to waste valuable nutrients, right? So increasing the surface area of the small intestine is very, very important. So the inner lining of the small intestine is covered with what are called villi. Okay, and villi, once again, are finger-like projections. Okay, and that's what we're seeing here in this cutaway histology microscopic image. Okay, these are villi, finger-like projections that, as you can see, increase contact surface area for all of the nutrients passing by this tiny little segment of the small intestine. The more surface area you have, the more opportunity to bind to nutrients there is. Okay, so know that this slide here is asking you, when it says name the part of the small intestine, know that these are the villi. Continuing, we also have ridges in the, see, you can see on this illustration, the pancreatic duct looks a lot more like fish bones, like I told you. Okay, all right, but back to the small intestine, and specifically this is the duodenal section of it. We have the villi that we talked about. Here we don't see the villi, but we see what are called the plicae circularis. Okay, these are just ridges of the small intestine. And again, these are folds, you know, again, like the rugae of the stomach. But in this case, it's not to allow the small intestine to get larger like the stomach does. But in this case, it's to increase surface area again, to again, try to latch onto and capture as many of the nutrients as we can coming through our alimentary canal. All right, so know the plicae circularis and know this image, because this looks a lot like a quiz question, because it is. All right, we're almost done, guys. Anatomy of the large intestine. So now we've made it through our 3 segments and the inner composition of the small intestine. Now we're to the large intestine. And here again, the anatomists, for the most part, did us a favor in naming it with logical names per segment, okay? And I'm going to emphasize this to you guys, okay? really, really try your best to remember when you're asked and you're asked to identify a given section of the large intestine or colon, keep in mind the direction the chyme is flowing. And that will help you to not get confused and remember, because if you consider the direction the chyme is moving as it exits the body, it'll help you to understand what segment you're looking at, okay? For example, we have here, the small intestine has been removed, but it would articulate right here with this yellow segment that is called the cecum, okay? And of course, that last section of the small intestine is called the ileum. So the valve, here it's actually called a valve. It's called the ileocecal valve. We'll talk more about that in lecture. But the ileum and the cecum come together at the ileocecal valve, okay? And this tiny little segment, the first segment, if you will, if you're chyme making your way out of the body, will be the cecum. And it's off of the cecum that this tiny little tail-like projection exists that is our appendix, okay? From the cecum, following the path of the chyme, like I highly recommend you do, we're going to ascend, right? It's going to go upward as it makes its way out of the body through the ascending colon. Then we're going to make a turn and we're going to transverse. To transverse means to go laterally, okay? We're going to go across laterally, horizontally, across the abdominal cavity. Then we're going to make another turn if we're that chyme on our way out of the body, and we're going to descend in the descending colon segment. And then we have a unique one like the cecum where we get this little J shape at the bottom. It's called the sigmoid colon. We labeled that. We drew in the arrow for that over on our drawing, on our study guide. And then the sigmoid colon ceases to become the colon or large intestine at that point, but continues as the alimentary canal, becoming the rectum and eventually the anus for excretion, okay? So cecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, anus, okay? All right, so for your lab quiz here, please pay attention to the parts that I had you label on your study guide. And those were the ascending colon, the transverse colon, the descending colon, and the sigmoid colon, okay? Those were the big ones that I told you to really to put down and know on your labeling, okay? So that's why. All right. So, and again, I'm just going to say it one more time, because inevitably, and for some reason, I don't know why people don't hear me when I say this, but they fail to remember what I said. Continue to, when you're analyzing and you're looking at the quiz and it says, name this segment here, consider the flow pattern of the chyme as it exits the body, and you will know that this is descending, okay? I get a lot of students, and that's the reason why I'm really emphasizing this. A lot of students label this side over here as the ascending and this side as the descending, okay? Because they're not remembering that very, very simple study tip. Pathway of the chyme, and you'll remember whether it's going up, ascending, or going down, descending. All right, and then our large intestines. Again, by the time chyme, waste at this point, has made it through the small intestine into the large intestine, we have absorbed a lot of water out of that chyme. We've also pulled a lot of those, ideally, most all of those monomers out of the chyme. So that chyme has gotten significantly drier and thicker, okay? So the large intestine / colon has these bands of significant muscle magnitude, because whereas the small intestine and the rest of the alimentary canal, via peristalsis only needed to squeeze with a moderate amount of force. Our large intestine, colon, because it's a much thicker paste that's much drier, needs a lot stronger muscular contractions to continue peristalsis and move that waste product to eventually excrete it, okay? So each of these muscular bands that we can see here of the large intestine, each of these are called haustra, okay? So know for the lab quiz that what this is asking you to identify, each of these bands is called a haustra, a muscular band of the large intestine called haustra. And again, we'll be talking more about that on Thursday, to be sure. All right, if you remember, back when I previewed the week before spring break, 2 weeks from today, back on March 3rd, we talked about the respiratory system and I previewed the lab quiz for you, right? And I said that it's going to be a combination quiz, lab quiz 3, of both the respiratory system and the digestive system. But there were also going to be a couple of unique categories, and I gave you the percentages for them, remember? And when we talked about respiratory, we said that a part of it was spirometry, right? And respiratory function testing, and what do the results of a spirometer test look like, right? And then I said today, when we got to digestive system, we were going to talk a little bit about enzymes. There's only a couple of questions related to enzymes, and then for whatever reason, the powers that be wanted us to embed a couple of clinical research questions. So there's literally only a couple questions on enzymes and a couple of questions on research. So that's what we're going to wrap up the lab here talking about today, okay? And then I'm going to recap the lab quiz for you, so stick with me. We're almost done. So enzymes, remember we've introduced enzymes in lecture today. They are a chemical catalyst. They're a chemical that starts a chemical reaction or intensifies a chemical reaction, okay? So know what an enzyme is, okay? By the time you take this lab quiz a week from today, you should have heard this enough times that this will be a given and a very obvious answer to you, okay? An enzyme is a catalyst. It's something that increases the speed or the intensity of a chemical reaction. All right, there are a couple of factors that influence the intensity of an enzyme, a reaction that an enzyme initiates, okay? And those are, the first one's pretty obvious, concentration, right? If you have more of a given enzyme, let's talk about amylase in the oral cavity, since that's the one we've talked about so far at pretty good length over in lecture. Amylase in our saliva. If there's more amylase in our saliva, we're going to have more effective chemical digestion. We're going to have a more intense chemical reaction than if we had less amylase. I think that's kind of common sense, right? And then, like so many things, pH, the acid versus alkaline ratio, and the temperature where the chemical reaction is occurring factor into how intense the chemical reaction will be. So know these three, know what an enzyme is, and know these three factors as influencers of the intensity of the chemical reaction. That's it for enzymes. I told you, 2 questions. I gave them to you, so you know them. I'm going to do the exact same with research, okay? I am a former chief of clinical research, and I had a passion for cardiovascular clinical research back in the day when I was younger and kind of new in the cardiology field. And so I spent a lot of years in doing clinical research, and I have, I think, 75 to 100 published articles out there that are being used to teach worldwide, and I've lectured all around the world on my findings. So it's been a great privilege. Clinical research has been tremendous to me. I love it. It seems, from my experience, that people, once you get out into the healthcare field, that people either are drawn to it or they repel it with all their might. It's either a love-hate relationship, and I totally understand that. That's fine. Either it's for you or it's not, okay? But scientific method is kind of the process by which clinical research evolves and occurs, okay? So I'm going to use a real basic example that's really obvious, okay? But let's say, I have this epiphany, this idea that I can contribute something to science. And my brainstorm is, again, this is going to be so obvious that it's ridiculous, but if a person gets on a treadmill and exercises on the treadmill, their heart rate will go up when they're exercising, okay? So scientific method would start with that idea. I'm going to go out, if I have this idea and I think it's novel and can contribute to advancing the knowledge in the field, I'm going to then start researching and find out if it's already been done. Because it's already been done, there's no sense in doing it. But if it's new and it hasn't been done or hasn't been done as thoroughly as it could be, we now have a topic area. So we have a project, right? So then we're going to develop the hypothesis. And the hypothesis, really in its most simple form, is an educated guess, okay? It's a relationship. It's what I described. It's an if-then. If the patient gets on the treadmill and starts to walk, then their heart rate will increase, okay? So that's the hypothesis. What you can't see here, it's cut out, is the experiment. Okay, we're going to experiment. We're going to put subjects on the treadmill and see if, in fact, this occurs. Okay, we're going to test our hypothesis. Then we're going to, of course, analyze our data statistically to find out if it's relevant. Is it extremely likely to be the reason? Because there are other reasons that could cause the heart rate to increase. You could have drank 32 ounces of coffee before you got on the treadmill. So we have to run a number of patients through and analyze the data to make sure that it's sound, that our hypothesis is actually true. Okay. And then ultimately, like I said, then we're going to publish and report our conclusions. So that's kind of an overview of what scientific method is. It's just a process that's pretty common sense, and many of you have been exposed to already in your lifetimes in one form or another. So what I want you to know from that and about clinical research for the lab quiz, again, there's only a couple of questions, is one, back to what is a hypothesis that we just talked about. It's that relationship between variables, the if-then statement. In my illustration, if a patient gets on a treadmill and we turn it on and make them start walking, then their heart rate is going to increase, okay? All right, so that's what a hypothesis is. Usually, guys, everyone does really good on these questions because I'm giving them to you, and they're really obvious, pretty introductory type terms. The second thing I want to talk about that I know you guys have probably heard about is an experimental group versus a control group. And just like the names say, they kind of describe what's going on. And I've done a lot of research in the pharmaceutical world over the years in the cardiovascular arena, and where we had a new drug, for example, let's say to correct some type of an arrhythmia. Okay, Pfizer comes up with a new drug and they need to test 100,000 patients to determine if, in fact, this drug works before they can pass FDA approval and release it to the market. Okay, so we're going to have the experimental group that in this illustration is going to actually receive the drug. Okay, and then we're going to monitor them and their arrhythmia in my illustration for the next 6 months or a year or 3 years maybe. Okay, to find out if that arrhythmia, in fact, got better or not. And then at the same time, we're going to have a control group, and neither group's going to know which is which. Okay, the control group's going to get the experimental group is going to get the actual pill, using my example, and the control group is going to get what looks like the actual pill, but it's just going to be a sugar tablet. There's not going to be any drug in it whatsoever. So that's why they're a control. They're a comparison group. okay? We wouldn't expect there to be any changes in this control group, right? Because they don't have the drug. At the end of 3 years, if the drug is working, this experimental group should show a tremendous improvement, whereas the control group really technically should show no improvement because they've been eating sugar every morning instead of a new pharmaceutical. Okay? So just know the difference between an experimental group versus a control group, specifically knowing what a control group is. It's just a comparison group against that experimental group that you would expect there to be no changes in. Okay? All right. That's it for the content, but don't go anywhere, okay? Don't go anywhere. I see it down to 32 of you now, okay? I want a real quick recap, okay, on the lab quiz number 3, because 1 week from today is lab quiz 3. We, of course, will be meeting for lecture on Thursday and on that Tuesday. So you're going to get reminders that that is your lab quiz number 3 day, 1 week from today, Tuesday. As always, open 8 a.m. to 8 p.m. The content you guys know from your calendar and what I've shared multiple times is on the respiratory system, and along with what we learned today, digestive system, along with scientific method and enzymes. Okay. About half of the quiz is on digestive versus respiratory, with just a few questions on spirometry, a few questions on scientific method that we just went over, and a few questions on enzymes that we just went over. And what I want to do here, because I've had a bunch of people start to cut off and leave during lab, is I'm going to give you guys a special opportunity for those of you that stuck with me today. Reward goes to those who comply and follow the course rules. What I'd like, I know you guys have emailed me already, and you've already emailed me with your lecture extra credit attendance, okay? This one isn't an attendance verification because, as I told you, I take attendance in lecture because we meet back to back, but not in lab. But right after we quit, in a minute here, when we stop lab, if you guys send me an email, another email that just simply says the word lab, just send lab to me, either as the subject or in the body. I'm going to award you guys for sticking around compared to the other almost half of the class that bailed out on us. I'm going to award you guys 2 extra credit points on the lab quiz. Okay? So again, I think that good behavior and following the rules should be rewarded. So I want to award you guys extra credit for sticking till the very end of the lab today versus about half the class that decided to leave early. So drop me another email right at the end here, just saying the words lab, and you will get some extra credit for having followed the rules and stuck here with me. Okay? Are there any questions on the lab before we break for today? Anyone that doesn't have questions, you're free to leave. Okay? Drop me that email.
Dr. Fine, I just wanted
Thank
to
you, Doc.
from my other classes, we have to start saving stuff from D2L because apparently some of the stuff is going to be taken off. So I just wanted to find out if that's going to also be affecting you. I know someone else in my maths class said with their lecture and lab, they're having to save some of the stuff.
Yes, that's a very good question, and it all has to do with federal compliance. We're being reviewed this month, and all of us professors, I had to have all of my faculty, I had to teach each of them to go in and clean up and make their things accessible for people that are visually impaired or have any other learning adjustments. It's an accessibility audit is what's occurring. It happens at all colleges. So for those that absolutely couldn't get their documents to be compliant, they were told to at least temporarily remove them so that we can pass, okay? So this class, you guys are lucky because this is an OER class. the first of its type. So all of my materials are compliant. So all of your materials are staying put in this class, both lecture and lab.
Okay, so we don't have to rush and save everything today. Okay.
You don't have to do anything for this class.
Okay, perfect. Thank you.
Fantastic. Thank you, Doug.
You're very welcome. I'm glad you brought that up. I'm very glad you brought that up. Okay. Okay. And any other questions, content or otherwise? And again, we'll see you guys on Thursday for lecture and Tuesday for lecture.
Sir, the last slide, in that it says like a control group. So I was just wondering, like, is it also called as a placebo treatment?
Yes, absolutely. Absolutely the same thing. It can be called a control group or a placebo group. Control group's a little more broad in terminology because a control group can affect like exercise stress testing, whereas placebo is typically drug only. But they're synonyms. They're two words for the same thing.
Okay, thank you.
You're welcome. All right, guys. Have an excellent rest of your afternoon. Drop me that email. I'll get you guys your extra credit points, and I'll see you guys Thursday at our normal time for lecture.
Thank you. Thanks.
Have a good day.
Okay, bye everyone. You guys as well.